And yes, I am the person between you and your dinner. So excuse me about that. So today I will talk about regenerative medicine. And this lecture has been divided into three parts, and today I will only focus about the cell-based system, that is the cell which is used for the use as a regenerative medicine. In that, a specific example, that is adipose-derived, you can say, stem cell, will be discussed today lecture. So when I consider about the regenerative and degenerative process, this picture is self-sufficient to explain everything. Basically, the degeneration means we are losing the tissue as well as, you can say, certain kind of the cells, and that we don't want to lose, but due to the aging and certain kind of the disease, we cannot stop you can say losing the cell and the extracellular matrix. So definitely we are going to lose our vital activity also. So regenerative process is that to heal with the, you can say, the cell, and basically the healing is not just providing certain symptomatic medicine, but it has to be improved the number of the cell inside the tissue to strengthen the tissue as well as organ. But the last picture you can see, it's really long way to achieve the youth from the degenerated phenomena. So in a simple way, that degenerative disease, which is very important in our domain, that disease with the function or structure of you can say affected tissue or organ change or worse over the time. Basically, the degenerative phenomena is highly linked with the aging as well as some extrinsic and intrinsic factors also. But the aging is the major culprit. Many degenerative diseases exist in some or related to the aging. And also sometime, main or you can say partial cause behind such disease is genetic. The most important thing is that some of the viruses and poison, or you can say some kind of the chemicals, that also cause the degenerative phenomena in our body. So we have to regenerate our body. That is the major task while we are treating the patient who have the degenerative feature. So in that case, basically, we have three approach. Number one, that is regenerate. So regenerate, identify the cause of the cause that can allow for the regeneration without, you can say, providing any kind of this care to the tissue or organ or wherever. My example is, you can say growing a new limb, especially in case of the very lower vertebrate, but not in a higher one. Repair is very important phenomena where we have to stimulate the tissue or cell or molecular level, that even at the level of the DNA, as you already saw in a first presentation, that how you can say certain kind of the pathway trigger the DNA based apoptosis. Similarly, here we can induce the you can say stimulate the DNA to repair the cell itself, and cell resident cell must transform into stem cell to provide the specific kind of the tissue which we need to heal the tissue or organ. Then another one is your replace. So replacement therapy is very important. It's a biological substitute is critical. That is very important. That biological substitute is critical in the lab that can implanted or replace the tissue or organ of interest. So right now, a lot of disease can be cured using the replacement therapy. And nowadays, instead of the cell tissue replacement, we are also looking for the epigenetic or certain kind of the molecule mediated replacement therapy. So there are several types of the regenerative you can say medicines or therapies are available right now, and some are still under clinical trials. Some have been adopted and proven, as well as, you can say, approved from the FDA. So cell-based therapy is very common, and among cell-based therapy, most frequently used therapy is PRP. You know very well that plasma-rich, platelet-rich plasma. So cell therapy, so therapy using the patient's own cell. Basically, right now there are you can say a high chances for immunogenic response from the you can say arthrograft. However, the autografting is very commonly used, and its effect is also very good. Another one is your therapy using the cell from the another person, like a blood transfusion, or you can say bone marrow transfusion, as well as you can say kidney and other tissue transplant. So obviously this can be happen, but the only thing is that the chances of the immunogenic rejection is very high. And another one, you can say the tissue engineering that we will learn in the second phase of our lecture, the second lecture. And other, you can say biomedical, biologics, and secretomes and gene therapy, that will be the second and third part of the second and third lecture. So in this picture, you can see that the overall summary about the this regenerative medicine. We can have the cytokine, which can obviously regulate the cell pathway or cell signaling. Then growth factors. There are several growth factors, and definitely these growth factors direct the you can say stem cell to get converted into specific progenitor cells, and that could be converted into a specific cell or tissue. And another one is that secretomes, that is very important, especially you can say microvessels, extracellular vesicle, that is very important. It contains DNA, RNA, and a certain kind of the protein marker, which is, all these DNA and the small miRNA, non-coding mRNAs, that is encapsulated within the lipid bilayer. And that is the most powerful tool to epigenetically control the cellular behavior. So here, that is very important. The stem cell therapy, it is not very new concept. People are using stem cell therapy. It's very ancient. The first stem cell therapy was bone marrow transplant, when the bone marrow was, you can say, provided to the patient from the congenital, you can say, identical twin to the patient, and the patient get recovered. So obviously, it is very important. So cell-based therapy, they could be provided in abundant quantity. That is very important. So we can provide millions to billions of the stem cell. They could be differentiated into many cell lineages in a regulated and reproducible way. Another one that could be, you can say, harvested by minimally invasive technique. So that's the most you can say promising thing while using the cell-based therapy. They could be safely and effectively be used in either an autologous or you can say allogenic host. Some allogenic cell-based therapy is already established. They could be you can say produced according to the current good manufacturing process. So there is not a big challenge to produce cell for the treatment. You know very well that in case of the stem cell, we can easily classify the stem cell based therapy into 6 types of the stem cell. One is embryonic stem cell, that usually called ESC. Then tissue specific that stem cell. Then you can say mesenchymal stem cell. The mesenchymal stem cell is you can say most favorable stem cell of the most of the researchers, because this you can say isolation and purification of mesenchymal stem cell is much more easier than other type of the cell line, and it can give so many type of the progenitor type of the cells. So when I talk about the mesenchymal, so you can see that, and we are very much interested in mesenchymal because our department is related to the orthopedic injuries, as well as you can say muscular orthopedic system. So we are interested in healing of the orthopedic injury. That's why we prefer the mesenchymal stem cell. Among the mesenchymal stem cells, we selected the adipose-derived, you can say, adipose-derived stem cell. So you may see that this tissue-specific, especially we are looking for the adipose-derived stem cell that is that can be go for the different type of the you can say target. So however, if you see that especially the adipose derived stem cell can produce the different type of the smooth muscles, as well as you can say other muscles, osteogenic muscles. So that's why this could be a very important tool for the for the developing the cell-based therapy for the especially degenerative musculoskeletal diseases. You can see that the mesenchymal stem cell have capability to you can say reconstruct hair, then heart repair, then also you can say the muscle regeneration and cartilage, ligament, and also we found certain evidence of with the tendons, then regeneration of the bone tissue. So obviously we are interested in this particular phenomena in our department, and that's why we focus over the adipose-derived stem cell. So you may see that any kind of the mesoderm is very, you can see that the mesoderm can give different type of the cell line. One is mesenchymal cell, another one is your hemopoietic cell. Hemopoietic further divided into common lymphoid and common myeloid. So we are not interested in these two. However, we are interested in only mesenchymal stem cell. Among the mesenchymal stem cell, we have the adipocyte, and this adipose derived stem cell can give all three you can say cell lineage which we are interested, like osteoblast, you can say chondroblast, and myoblast. So this will be you can say starting tool for our orthopedic department. Here some study is suggesting that why you can say adipose derived stem cell so important, and how it can play an important role in post surgery you can say wound management. So there is so many evidence that post surgery you can say rapid healing, or you can say that appropriate healing is achieved while we provided certain kind of the cell-based therapy. So these certain papers you can see that these are the you can say providing insights about how to isolate and how to use the adipose derived cells for the you can say for the treatment of the musculoskeletal disease. Now come to the adipose tissue. Adipose tissue is the most abundant kind of the most frequently found tissue in our body. And it is mainly composed of the adipocyte, that is about 90% of the tissue volume, and stromal vascular fraction, including the fibroblast, endothelial cells, and you can say pre-adipocyte, vascular smooth muscle, lymphocyte, and resident monocyte macrophages, etc. are there. So the adipose adipose stem cell have you can say mesodermal origin, but they have the potential to differentiate into several lineage, especially you can say osteogenic, chondrogenic, and adipogenic, you can say cardiomyogenic, myogenic, and neurogenic cells. However, their differentiation required certain kind of the stimulant, and that stimulant we will see in the next couple of slides. So they can be differentiated into tissue of the endo as well as you can say ectodermal lineage, and such as hepatocyte, pancreatic islets cells, endothelial cells, neural cells, and epithelial cells too. It was shown that adipose stem cell harvested from the superficial abdominal region are significantly more resistant to apoptosis than the other part. So, and we know very well that the in case of the abdominal region, the adipose tissue amount is very high. So harvesting is not a big challenge from even for the, you can say autografting or you can say allografting. So it is very important that when you see the stem cell which is present in a stromal vascular fraction, especially isolated from the adipose tissue. So we took the adipose tissue over here, and it's usually mechanical or you can say enzymatical digestion. But in our laboratory, we prefer enzymatic digestion. So after enzymatic digestion, we just centrifuge it, and after centrifugation, we have three layers. You can see that the fat layer, then we have the watery layer, and then another one is your SVF. This SVF contain at least 4 types of these cells, and these cells are pre-adipocyte, which is predominantly, you can say, express the marker CD34 plus and CD31 minus. Then you can say mature endothelial cells that also have CD31 plus and CD34 plus, then mature endothelial cell which have predominant marker of the CD31 plus, you can say, then pericytes that have some different marker like CD146 plus. That's very important to know that what kind of the resident cells found in SVF. So adipose-derived stem cells are, you can say, a subpopulation of adult mesenchymal stem cells, and it can isolate from the adipose tissue. It doesn't matter. I mean, you can isolate from brown adipose as well as the white adipose. However, the property, if you can say, adipose-derived stem cell from the brown and white is little different. Like white adipose tissue, it's a subcutaneous and visceral, you can say, visceral distribution, act as an energy storage source, while brown is mainly in a neonates in the neck, you can say, media sternum, and you can say, carpicular region involving in the thermogenin mediated heat production. It's a kind of the function. So adipose-derived stem cell obtained from the white and brown, that is the, you can say, adipose tissue, differ in their property. That white adipose progenitor do not express myogenic regulatory factor, while those brown, it express the myogenic, you can say, regulatory factor. So this is the, you can say, fundamental difference between the adipose derived stem cell from the white and brown, you can say, adipose tissue. So isolation of the adipose, you can say, derived stem cell is possible from the, you can say, mechanico-chemical digestion of the adipose tissue. And finally, we will have the stromal vascular fraction. So this, you can say, the expression of the marker varies, you can say, based on the donor, cell culture, passage, and cryopreservation, you can say, duration and methodology. So adipose tissue sample are generally harvested by 4 methodology, which is commonly used depending on the facilities available in respective hospitals. So like Coleman's technique, which is then surgical resections, then power assisted liposuction, then laser assisted liposuctions. This 4 type of the commonly used technique for you can say, harvesting the adipose tissue in hospital. So the most popular one is the Coleman techniques, because it consists of the 3 steps. One is the low pressure lipo, you can say, lipo aspiration, then centrifugation at 3000 RPM, then collection and re-injection of the highly enriched cell deposit layer into the grafted site where we need to inject over there. The liposuction provide a tiny pieces of the, you can say, adipose tissue in a form of the lipoaspirate with no need for extra mechanical manipulation. In case of the evidence illustrated that surgical procedure gave large number of the adipose derived stem cell and higher, you can say, collagenicity, while those isolated by the, you can say, pressurized liposuction, or you can say laser guided liposuction. So it means the surgical method is much better while you are isolating the you can say adipose trying to isolate the adipose derived stem cell. So another one is that the acquired adipose tissue could be used as a fresh, or you can store it up to 2 years under a properly, you can say, cryopreserved protocol. So without any negative effect of on adipose derived stem cell yield, survival, and stamina. So this is advantage of the adipose tissue, and in any surgical process we can easily get a good amount of the adipose you can say tissue. So isolating the adipose derived stem cell is much more easier than any isolation of the any other stem cell. So adipose tissue in a protocol, we just took the small pieces. It washed several times. The washing is very important because we have to remove all the hemopoietic content from the tissue. So tissue must have only adipose, you can say tissue fraction, adipose fraction. So after several washing, we have to digest the tissue with the collagenase. The collagenase digestion is very important to separate the cells from the extracellular matrix, and then it has to be centrifuged. The rest of the supernatant we don't need, and collect the pellet. The pellet have most of the cells. It's just like SBF. So collect the pellet and incubate this pellet in a media. The media must have the media. Usually we prefer the media like DMEM, high glucose media with the 10% FBS supplement, the fetus bovine, you can say serum supplement. This is very important, and we need to change the media, you can say after 48 hours to provide any kind of the, you can say additional shock from during the process of this culture condition. So the isolation is very important, and the isolation you can isolate this is the one of the protocol which we follow in our laboratory, that isolation of the adipose from the two method. One is that resected adipose tissue that in generally I will call RAT, and another one is your lipoaspirates, that liposuction mediated you can say tissue is there. So first of all, we have to cut into small pieces and plated directly on culture flask, and then you can say turning the culture flask. I mean, you have to make proper shearing this culture flask in every 12 hours for the 48 to 72 hours, to allow the plastic adherence of the cell to crawl from the pieces. So from the pieces, the cells should become outside, and this is very important. So another method is to treat the pieces with the collagenase. In our laboratory, we use first we use collagenase to digest the tissue, and then isolate the SBF pellet, and then this SBF pellet has been you can say cultured separately as well as co-cultured with the RAT pieces in a certain flask. So this is you can say preparation of the tissue, and later on we have to understand that the media which we are using, that DME high glucose media, usually the concentration is 4.5 gram per liter. With the 2% you can say L-glutamine, that is very important content, and then we use the 10% of FBS. That's very important because it contains all kind of the required nutrition for the cell growth and you can say proliferation. Then we use the you can say antibiotic solution, that is 1%. It's penicillin and streptomycin mixture is there to avoid any kind of bacterial contamination. Then similarly, we use the similar protocol for the using the liposuction aspirates, and same after certain culture we got you can say find out the cells, and the cells has to be finally go with the density of the 2.6 into 10 to the power 5 for per centimeter square plate should be inoculated with there, and final concentration of the finally it has to be incubated into 5% CO2 at 37 degrees Celsius. After 48 hours the culture flask will be washed and with the PBS and remove non-adherent cells, and then the cells will be harvested further for the different making the different passage lines. So you can see that how it look like. It's a very simple way that we got the tissue. You can see that in a picture A, that the tissue we have, then this tissue has to be treated properly. I mean, after washing, we just got the fat layer, and then lower you can get the blood or aqueous layer. And you may find that the cells, when we inoculated into plate, the flask, the cells are very limited, and then it started the growth over there. Then after growth, it gets differentiated, and then we harvested over there. This is the one of the lipoaspirate procedure, and this procedure is very you can say right now most of the hospitals, they use the automated process where they don't use the needle based, while you can say under developing countries or underdeveloped countries, they still use the needle mechanism for isolating the adipose from the belly region, or you can say abdominal region. So this is how you can see the figure 3.11 how the adipose tissue after liposuction can look like. So this is all about, procedure that harvest the cells and store over there. And after restoration, it should immediately go to the laboratory for further, you can say, proceedings. And then we go for the centrifuge. And after centrifuge, we get the oil, and we remove every time the oil. And after removal of the oil, we will have the two phases. One is adipose phase, another one is your SBF phase at the bottom. So you may see that how finally at the figure 3.19 that compact adipose tissue, it should be transferred into sterile 50 ml tube and weigh out how much it is there for further evaluation. Then it's isolation process under, you can say, standard condition. And then we just get after centrifugation that we can, this is the entire media with the tissue, and then centrifuge it. After centrifugation, we can get here that pellet. The pellet itself have only adipocytes and adipose derived stem cell. So that will be used for our further study. Now you may ask a question, how you will know that you will have the adipose stem cell in an entire your population? Yes, the entire cell population is not adipose derived, stem cell. That's why it is very important to know that what is exact concentration of adipose derived stem cell present in our culture. So only, the available method, that is the flow cytometric method, and by that we can find out the reported marker which is present over the adipose stem cell. And this marker from the total population of the cell, it gives an idea that how much percentage of these cells are adipose derived stem cell, and that will give an insight that what concentration we need to provide the patient for appropriate healing. So in very common phenomena that there is a big question why we need a regenerative medicines in a surgery. Basically, when we talk about the degenerative diseases, we know very well that surgery can help a lot, but we need to provide the certain kind of the grafting. It's either allograft and autograft. You can say some kind of the mimic of extracellular matrix to provide the strength to the certain degenerated area. So it has been observed that if we provide after surgery that grafted area, if you provide the cell treatment, you can say cell mediated biologics, the healing is very, you can say, rapid, and obviously the resident cell number is increasing very fast. So it is very important that regenerative medicine and surgery is rapidly, expanding the branches of the translational research in tissue engineering, cellular and molecular biology. So as adipose tissue is easy, you can say, accessible and abundant in a source of adult stem cell and can differentiate along multiple lineage, it can be considered as a good candidate in aesthetic medicine. So that's why we selected the adipose tissue for our study, as well as targeting adipose tissue for further study. The another one is that regenerative potential of the adipose derived stem cell has been demonstrated for several years with a technique such as fat grafting or replacement of the soft tissue. So we have a concrete evidence that yes, by any mean, adipose tissue is used in a surgery, especially for the plastic surgery and wound healing. It is used for the long time. So you may see this particular table will provide a proper insight to you that soft application of the adipose tissue, like soft tissue reconstruction, like breast augmentation, that facial lipotrophy, then facial augmentation, and these are the some evidence-based study which has been provided as a source article. Then bony reconstruction, then wound healing. It has been found that in case of the radiation atrophy, and it's a very adipose derived tissue play a very important role over there. So in totality, when we say that the in case of the translational medicine, it is very important to make a balance between the, you can say, clinics, industries, and research. And translation medicine is like adipose derived tissue is very important because a lot of pharma companies as well as hospitals are focusing over there. And when we say that they, that uh, when we try to tissue you transplant or any kind of the soft tissue transplant, so at the moment there are so many type of the factor which don't allow to heal properly, and that's why stem cell play a very important role, because we can guide them to differentiate a specific type of the lineage, and obviously that kind of the progenitor cells can repair the tissue as well as, you can say, organ by themselves, because it's a biological phenomena. Once the number of the you can say adequate number of the stem cells are there and differentiation occurs, so obviously healing is very faster. So you may see that major cause of the transplanted, you can say, cell death, that shear stress, that host inflammatory response, and low, you can say, nutrient supply, hypoxia, so many things are there. While this kind of the problems are usually overcome by the surgery along with the, you can say, cell-based therapy. So obviously it will give a more, flexible way to heal the wound, as well as, you can say, degenerated condition. Now, you can see that this is, though here the references are not provided, but it has been proven that if we have the adipose-derived stem cell, that can make so many things. For example, if you see the, it can go for the aging repair, it can production of the young cells is possible, and how we can regulate that. There are certain kind of the cytokines as well as the growth factors that play a very vital role. For example, the combination of IL-6 and TGF-beta-6 along with the TLR2 and TLR4, GDF, that is the different type of growth differentiation factor, and other growth factors. If we provide a combination, then it could be converted into macrophages. And if the macrophages is again, this macrophages can produce different type of the cytokine and other growth factors that again make this type of the cyclic over there. You can see that if we look for the ECM, so in this case, if ECM and TGF-beta is there, that can be guided by the GDF11 and other growth factors. It could be converted to fibroblast. This fibroblast can be converted to different type of the cells. Similarly, you can see that it can be converted to fibroblast, then platelets, and obviously it can help in blood clot formation, hemostasis, and etc. So obviously, in totality, if you see in case of the wound healing, they can promote the angiogenesis, and the angiogenesis is very important phenomena for any kind of the tissue regeneration, because it combats with the hypoxia. It also provide the proper nutrient to the developing cells, and it also allow a proper hormonal as well as, you can say, growth factors supplement to the injured area to repair as well as remodel the entire tissue. So this is very important that endobronchial administration of the adipose-derived stem cell precursor was demonstrated safe in previous study, that you can find the reference, that the active role of the patient could support therapy effect during the 3 phase of the breathing, inspiration, holding, and expiration. So obviously, and also another researcher, practitioner, that reported that adipose-derived stem cell differentiate into insulin producing cell in culture and normalized, you can say, beta cell. Then adipose-derived stem cell, in case of the embryonic stem cell, have been examined for treatment of the cutaneous wound and also blood glucose level after the transplantation into diabetic mice. So yes, this is the potential. That's why we selected the adipose-derived tissue, because once we are trying to heal any tissue, then not only a single type of the cell is required. There are so many type of the cells is required for proper homeostasis of the tissue. So you may find that the adipose-derived tissue can give melanocyte, adipocyte, chondroblast, osteoblast, neuron, fibroblast, endothelial cell, muscle cell. That different type of the cells can be possible. Only thing is that we need to provide the appropriate stimuli like a cytokine and growth hormone to give a proper direction to have the cell in a desirable manner. So you may ask that is it any protocol is already established? Yes, there are so many literature that suggests that how we can guide adipose-derived stem cell into a specific type of the cell. For example, there are so many, you can say, in vitro condition, like they use the media along with the certain kind of the stimuli like 0.4 millimole isobutyl methoxy ethane, and also insulin and idomethacin. So this compound can this particular protocol provide after 14 days the cells converted into adipocytes. So adipose-derived stem cell has been converted into adipocyte. Similar observation has been found with the certain different, type of the compound, which also provide, you can say, trigger the adipocyte development. So yes, there are so many type of the, you can say, stimulant are known, and by which we can produce the adipocyte in vitro, and that adipocyte can be re-implanted into the patient because it has been isolated from the patient itself. So obviously the autografting is not a problem. However, there are some paper also suggests that the allografting is not a big challenge nowadays. Now, if you want to make osteoblast, then you need to modify your media, and you just see that instead of the DMEM, we use the alpha MEM. And 10% FCS is there, then obviously DEX and ascorbic acid. Then another compound that is very important, it is your beta glycerophosphate. This beta glycerophosphate is an important, not only beta glycerophosphate, any kind of the phosphate you see here. This phosphate is important precursor for making any adipose-derived cell into converting adipose-derived cell into osteoblast, and then this osteoblast can be implant into the for the bone, you can say, regeneration. Similarly, chondrocyte, you know very well, the cartilage production is cartilage is one of the, high, you can say, hotcake in our medical science. So you just see that this particular adipose-derived cell can easily converted into chondrocyte. However, the protocol is very important in this. We need the phosphate. Along with that, we need TGF-beta3, and this TGF-beta3 and phosphate. Here we use the acidic phosphate instead of the, you just see that here we use dihydroxyphosphate, or you can say beta glycerophosphate most of the time, beta glycerophosphate. But in the case of the chondrocyte, the people use the phosphate in a form of the how you can say acidic form, phosphate is there. So similarly, you can see that ascorbate 2-phosphate. Instead of the glycero, they use the you can say inorganic type of the phosphate in a media, and this can provide you can say specific role like the protein synthesis and proteoglycan production. Similarly, it can be converted into chondrocyte. So there are so many type of the protocols are known, and yes, these protocols can easily adopt for development of the chondrocyte from the adipose-derived stem cell. The another one is the neuron development. This kind of the experiment has been also studied by so many researchers. However, that this DMEM N2 supplement and butylated hydroxyanisole and KCl is very important over there. So, and this protocol was very effective to produce the neurons. So not only this kind of the study also gave a hope for the neural, you can say, regeneration using the adipose-derived tissue. However, still a lot of optimization is required, but maybe in future we will have you can say the procedure to in vivo development of the neuron using the adipose derived stem cell implant. Then endothelial and also fibroblast, it can be possible. How you can see that a specific type of the growth factor is required for developing a different type of the cell type from the adipose derived tissue. The muscle cell is also again very important, and here it has been found that the hydrocortisone is very important, you can say key ingredient to protocol for making skeletal muscle. And in another protocol they found that the azacitidine is very important. However, the BMP is also found very crucial along with the TGF beta for the smooth muscle production. If you provide alpha MEM instead of the DMEM along with the angiostatin, it has been found that small muscle production is higher within the 4 days. In case of the DMEM with the certain other modification, you can find the smooth muscles and within 6 days, and their application can be used for the different purpose. So yes, you can guide adipose derived stem cell to convert into a specific type of the cell, and that cell can be used for the treatment, or directly you can in vivo you can guide the molecule using the certain type of the regulators. And keratinocyte, then cardiomyocyte. Similarly, the well-established protocols are there. And every year people are trying to develop a new protocol for, and obviously that protocol must be easy, so we can inject that kind of the stimulant inside at the specific site of the grafting. So obviously it will be, there are a lot of studies going on. So you may see that the production is not a big challenge using the adipose derived stem cell. So adipose, as we know that the adipose tissue is a worthy source of the mesenchymal stem cell, easily reached in several simple steps by the SVF formation, and stem cell purification and culture is very easy. So different harvesting and mechanical chemical purification techniques and isolation of the ADSCs in large quantities are possible. Then experiment-based data that illustrate, you can say, approving the proliferative rate without further specific supplementation to keep the adipose-derived stem cell growing. So yes, maintenance of the adipose-derived stem cell is not a big challenge in a laboratory. Genetic stability is very high. It has been found that the immunomodulatory feature, effective response to the chemical differentiation, inducers, and generating various specialized cell lineage support to the idea to use the adipose-derived stem cell in autologous as well as allogenic clinical cell therapies and tissue regeneration projects. And so many are, studies are under clinical trial, and some have been, passed the phase 1 as well as phase 2. So therefore, it would be useful to, you can say, take into consideration that applicability of the adipose-derived stem cell in the treatment of the relief of human disease and the subsequent adverse outcome is very important. Here, I just try to share very important feature, that clinical trial which is registered for the using the adipose derived stem cell, and I just use the filter that is used as a therapy directly. So you may see that there are so many adipose derived cell, and they are used for the hair regrowth, and you can say treatment of the perineal fistula, you can say Crohn's disease, some type of the keloids and diabetic foot. And I had a study of the diabetic foot also, but I didn't include it over here. And then adipose-derived stem cell versus platelet-rich plasma for follicular unit extraction. I mean, so many studies are going on, and you can understand that by this particular picture, why this study is so important. You just see that South Korea is very, important leader in using the clinical trial. You just see that Republic of Korea is having the 20 clinical trial using adipose-derived stem cell for the treatment of the different degenerative diseases. However, if you see the clinical trial, I mean, different type of the study, so you just see that the gastrointestinal, the 13. However, in our case, like musculoskeletal and that osteoarthritis, you just consider it is about 23 clinical trial is going on globally. So yes, the adipose-derived cells have a lot of future. And number of the trials using the specific route, especially, you know, local site is very important. As I say that we want to make a, you can say, patch specific or wound specific insertion of the stem cell to improve the wound healing as well as regenerate the tissue organ itself. You can see that the number of trial using specified adipose resources. So you may see that the adipose derived SVF is very high. However, autologous and is after that. A lot of people are also using the allogenic and allogenic cultured, allogenic differentiated, and for the developing as a normalized medicine, just like other biologics. So it could be, its price will be very cheap if it will be allogenic instead of the autogenic. So I must say thanks to all of you. Yes, I am between all of you, between you and your dinner. So enjoy your dinner. And I must thanks to my team and especially Professor No for providing this opportunity. And I hope you have any question. Please let me allow to answer your questions.
Thank you. Thank you very much. Very informative for your presentations. I have a question. One question about, my question is about the fate of the adipose stem cell. When we do cell-based stem cell therapy, at the time when we inject the adipose stem cell into the region site, at that time, which one is more important? The role of the stem cell in the niche. For example, the stem cell itself will differentiation to the target cell, or they will make a paracrine effect or something. What do you think about that?
Yeah, I mean, sir, the efficacy of the using a stem cell is not very high because there are two lacking. Number one, optimization of the stem cell is a major concern. Number two, the paracrine effect, as well as you can say, like you can see this picture is very important, that why the cell, transplanted cell have the death. You just see that the severe stress, host inflammatory response, host immune response, low nutrient supply, hypoxia, then oxidative stress, and that onakines is very important. But we can improve you can say efficacy of the stem cell. So we need to consider this following, factors like extracellular vesicular exosome supplements should be there. That means my second lecture, I will talk about why using the stem cell and that secretome is important for making the better response while using the stem cell. Then tissue cell conditioning is very important when we are injecting any stem cell to the localized area. Then cytokine modulation is very important because most of the time the cytokine is a culprit which don't allow to grow the specific stem cell, because stem cell is a proliferative cell. So definitely we need a specific, or you can say a guided type of the healing over there. So cytokine modulation is very important. Then extracellular, you can say vesicular exosome supplement is again play a very important role. Then ROS, as my first presentation, we already saw that the ROS is very important for inducing the apoptosis, or you can say cell death. So ROS, you can say management is that another phenomena. Then extracellular, that is the this extracellular can play this very important role. So that's why I will talk to in the next lecture. The extracellular vesicle is very important role and how it can help the stem cell. So must be give a specific type of the cell line at the applied area. So you can see that and another thing is that the complement inactivation should be there. So this type of the consideration enhance the acceptance of the stem cell and their survival, and finally differentiation and remodeling of the tissue. So it have three phase, and we have to provide and close monitoring of the three phase. That means the cell along with the certain biologics which can guide the stem cell into differentiation, and further remodeling should be there. So these are the important considerations.
Yeah, I agree with your opinion. So, in clinical field, when we inject the stem cell into the patient, but environment not good, so the cell, injected cell, cannot longer live there. So there are so many strips in there. So cell death occurred frequently there. So I think the Parkinson effect is more important to healing of the region site cell.
Yes.
So I recommend you to give us some two more lecture. So secretive of the article stem cell, and then exosome or microRNA or other things. So please give us good presentation next time. Thank you for your presentation. 혹시 질문이나 혹시 코멘트 있으신 분 계신가요? 그 비빅 박사가 되게 열정적으로 강의를 해주셔서 많이 배웠습니다. 근데 시간이 좀 많이 지난 관계로 오늘 연구에는 여기까지만 하도록 하겠습니다. 또 질문 있으신 분들 개인적으로 질문하시고요. 오늘 많이 참여해 주셔서 감사합니다. 다음 달에 또 뵙겠습니다. 감사합니다.
Thank you, sir.